So, animal development. I'll start you off with a minor quiz. What's an animal? Suggestions? This is a phylogenetic question. What is the synapomorphy, the common trait of all animals? Sam? Multicellular.
Heterotrophic.
Eats other things. I learned this. I learned those two, and I learned another one. Neither is correct. None of those are correct. Andrew had a suggestion.
Sexual reproduction.
Sexual reproduction is the third thing I learned when I did it. It's actually develops through a blastomere. Blastocoel's the whole. Blastula. Dear God, if I can't get that term right at the beginning of this, we're in trouble. But the other things help. You're going to be multicellular if you're developing through a blastula, and so forth. But that's it. But the other things are also happen to be true. They reproduce sexually, and they are heterotrophic. They eat other things. So when you do need to broaden your horizons when you think about what's an animal, though. Okay, but they share this developmental process. So for that reason, it's useful to look at lots of different things. Because lots of different things are animals. And today we're going to go from a vertebrate, which is fairly close to us, that's the chick, to something about as far out as you can get and still be an animal. We'll talk about one step further that we won't go. But that's a sea star. We're going to use slides of all these. So a sea star is just one step away from as far away as you could get, in that it's still a deuterostome. What's a deuterostome? And what's it contrast with? It contrasts with a protostome. And what that means is when you start to gastrulate, the hole that you make, when you start tucking the blastula into itself, the hole that you make from that becomes the butt instead of the mouth. That is the very practical and very technical definition of this. That's as far as it goes. This distinction has mattered a little less recently as we've gained a lot of molecular evidence to suggest how things are related. But it was one of the major ways that the early phylogenists grouped animals. They used this trait to mark one of the biggest branches in the animal phyla. So we are also deuterostomes. Our hole becomes the butt. We make a different mouth. Their development is relatively simple because they're going to form this free-swimming larva that only has so many structures, and you'll be able to observe those on your slides. We're going to cover and make you responsible for a lot of vocabulary, so if you see it as a keyword in your book or you see it in this presentation, it's there for a reason, and it is also therefore observable either on the slide of the sea star or in the slides of the chick. We won't quiz you on anything that you can't actually see. And that's how shallow we're staying in development, because there's a lot hidden from the eye. These are those terms. I think we'll get to them in subsequent slides. So, be able to draw these distinctions. Indeterminate versus determinate developmental stages. The name is informative. If it's determinate, it's already determined. It cannot be changed. If it's indeterminate, it can be changed. And this is one of those major phenomena of development. Let's see if I can rattle them off the top of my head. During development, you need to do several things. You need to grow. You need to have your cells divide. You need to specialize those cells to differentiate them or determine them. That's all the same thing. Cells need to move around compared to each other. They need to migrate. And they need to communicate with each other and tell them which cell to become. So there's communication between them. You mix and match most of those things, and you end up with a fully differentiated cell, tissue, organ, and ultimately individual. So development's cool because we get to figure out how to make all these different things happen, how to run that developmental program to get you into your mature state. Coming back to this term, indeterminate versus determinate, the very earliest cell divisions produce essentially identical cells. They are merely increasing the number of cells that are available to work with. This is Not strictly true throughout all development in all species, but certainly true in the most familiar cases, like us. So if you have a fertilized human egg, you are now, you have your complete genetic complement. If you divide that cell and actually physically separate it, this is how we end up with identical twins. They go on sometimes to even share a single placenta. Not always, but that's one way to tell them in utero that they're the same. This is different from having two different eggs be fertilized. That's fraternal twins. You need to know the distinction between that. An indeterminate cell in one of these early divisions, you could do this almost infinitely. Okay, you could keep plucking one cell off of the other after they divided, and you could come up with the correct programming of that cell to make an individual. What you would run out of is enough cellular mass and ingredients to actually have the cell be viable and set up all the support tissue that it needs, and so forth. Later divisions are going to take those same indeterminate ones and divide asymmetrically. There are molecular mechanisms for doing that. We're not going to quiz you on it, but if you want to chat about it, I'm very good at that. And ultimately, one cell is going to do one thing, and the other cell is going to do the other. Typically, what will happen is when one of these indeterminate cells divides, it will spin off a cell that will ultimately become more restricted in fate, while the other cell will remain more plastic, more able to, more indeterminate, that is. And so this is what popularly now is called stem cells. So a stem cell basically regenerates itself and creates a daughter cell that can go on to do some other function. So I think we can move on from that. Anyway, so we need to understand the structure of the gastrula. That's after we have enough cell divisions to get a nice ball of cells with a hole in it called the blastocele. That structure will fold in upon itself, and the layers that result from just the actual invagination of that, but also some of the cellular signaling that goes on in between the layers of cells as they move past each other, gives us 3 layers. And that's what we call triploblastic. It's common to almost all animals. There are some diploplastic animals, but you've probably seen in lecture those layers are ectoderm, mesoderm, endoderm. You're responsible for knowing what those different tissues develop into. Other terms when we're looking at this, archenteron is the new hole that gets made. The new hole that gets made as the cells start pushing inside. The existing hole, which is called the blastocele, that's spelled C-O-E-L at the end of it, should not be confused with the blastopore, which is essentially the end of the archenteron. A figure will help that, and I'm sure that'll come up. Otherwise, I'll insert it into the PowerPoint after the fact. Do know your different layers I already mentioned. The ectoderm, which it ends up on the outside of the gastrula, becomes the epidermis, and by signaling and folding, you get the nervous system as another tube inside of that. Both those structures are ectodermal. Mesoderm is anything sticky, including connective tissue and muscle. And the endoderm is basically anything that you classically think of as an organ, an internal organ. So lungs, guts, liver, pancreas, not the kidney actually, but just about anything that we opened up in the visceral cavity. So I already said blastopore becomes the butt. The notochord becomes your spinal column. We'll see that in the chick. That's kind of one of this streak of mesoderm that goes down the side, and somites are on the outside of that. Those become the actual individual vertebrae and the muscles associated with each vertebra, as well as degenerating into the intervertebral discs, which is something that nobody actually studies except the Children's Hospital, actually. When you're looking at, in particular, your C star embryos, you need to be able to distinguish the different stages that they're in based simply on their appearance. There should be a fertilized egg that shouldn't look like much. I think there's a nucleus that makes it more visible. Then you need to be able to decide, is this a 2-cell, 4-cell, 8-cell, 16-cell, based on how it looks. Those are actually the names of the stages we want you to learn. If it only has 8 cells, it's an 8-cell stage embryo. Okay? That means it's gone through 4 divisions. Division 1 went from 1 to 2, Division 2 went from 2 to 4, and Division 3 went from 4 to 8, so I apparently got it wrong already. Remember, it's exponential. So it's the 3rd division that got us to the 8-cell stage. Morula, Latin for raspberry, you already read in your manuals, is probably 2 or 3 more divisions after those stages that are numbered by the cells, and you'll see it looks like a raspberry or a mulberry. Once the cell's getting small enough and there's an actual hole in the embryo, we're going to call that a blastula. Okay? Once it starts folding on itself, this is very obvious in the C star, we'll call it a gastrula, and I don't think that we're really going to distinguish the later stages, but in our systems, the starfish is moving to what we would call the larva. Here are the pictures of they. So, again, the distinguishing characteristic of the actually, I take it back. This is the unfertilized stage. Only after fertilization do we get rid of that nice nucleus. Once fertilized, it looks like a single cell. Then it's just the number of cleavages that are visible. This is 2, this is 4, this is 4 from a different view. 8, 8, 16. And then morula really only lasts, like I said, a cell division or 2. I would not call this a morula. I think that's already a blastula. This little creeping in of tissue here is the very beginning of gastrulation. So the tissue on the outside is actually starting to tuck its way into the embryo. And that just gets more pronounced as time goes by. You see this additional layer of cells on the inside. This is getting more towards the actual larval stage. In particular, I had to look up one of your keywords, those coelemic cavities. That's actually their future mesoderm, which is done a lot differently than us, but that's actually these little pouches on the side here, so this is still a gastrula. But ultimately we're working towards a larva that's going to have the ability to eat and swim freely. So yeah, once you can see all the different folds on the inside, it's a larva. Use this slide and the previous one and memorize what's what, ask questions in lab. Okay, so we're also going to look at the vertebrate. In case you ever wonder what you cracked open for breakfast, this is the outline of that. There are many different membranes. I don't think necessarily we're going to get you on that. You should realize that the white part is albumin. That's protein food for the embryo. The yolk is the source of most of the calories. You know that from your nutrition classes and such. But also this is what the embryo is directly attached to. That's this small little fertilized bit here, the germinal disc or blastodisc. One of my favorite little tidbits, maybe something that you'll actually remember after you leave this class 5 years from now. What came first, the chicken or the egg? Well, we know it really wasn't that, it was the reptile, but the answer is still neither. The answer is, what do you need to survive in an encased environment like an egg? You can breathe a little bit, and that's fine, but what you can't do is sit there and soil your own house. Do you ever wonder why bird poop is solid? Or excuse me, not bird poop, bird pee, the white stuff when you hit your car. It's because if you're going to be living in an egg for 21 days, you have to be able to set your urea waste to the side without it being suspended in all the stuff that you want to do your normal biological chemistry in. You have to be able to actually separate it. and that's the function of having solid uric waste, is for life in the egg. That had to come first, not the chicken or the egg. I think it's interesting anyway. So again, you're responsible for anything we can actually visualize in the microscope. These things will get a little fuzzy. You're going to have to play with the plane, the focus that is, on your microscopes to get a lot of these structures quite right. But let's go through everything. Primitive streak is only important in chick biology, so that one I don't actually know that well, but know that it is mesodermal, the streak here. And it's associated with, actually I strike that, I'm ignorant on primitive streak, but you know where it is now because you can see it labeled here. The mesodermal components that we can actually see, I don't think we can actually visualize the notochord, but we can see the somites. These give us different things. The notochord, like I said, gives us the vertebral column, or it gives us the intervertebral disc. Moreover, it signals to help specify the neural tube on top of it. But the real functional unit here is the somite. So if you want to know how to make your different vertebrates, if you want to make a snake with 300 different vertebrae, or a human with 37 or 43 or whatever they're going to tell you we have, it's precisely this. It's the number of somites, these little chunks of mesoderm that we make, that determine that. And those give us different components. They give us bone, skeleton, bone for the skeleton, and muscle to move the skeleton. Anyway, moving on. You can see a spinal cord. That's why I waffled on the primitive streak. Connected to the various compartments in the hindbrain, midbrain, and forebrain. And so those have their own terms. If you're forebrain, you're prosencephalon. If you're midbrain, you're mes, middle encephalon. And then in the hindbrain, you're medencephalon. And the other term for that is the rhombencephalon. And the rhombencephalon you should be able to see as a bunch of bumps waving through the these neural tubes. This is actually a ventral view, meaning we're looking at it from the belly. You can see the heart loop will come out to the side. You'll see the very nasty looking vasculature attached to the egg yolk, the vitelline veins, come in here on the sides. And you'll just have to notice that these bumps coming off the side of the brain are actually the developing eyes. So that's the optic vesicle. There's one little slant that you can see, I hope it's on the next slide, of a more mature one where you can actually determine some of the endoderm. But this one's about, let's see, be able to identify the structures as they get older. You'll see that the forebrain, we're used to it being very big, but is really just this little lump here compared to the I don't know if I like that. The diencephalon's actually, I don't like that distinction at all. I'll get back to you on that terminology, but midbrain's up here, hindbrain is back here, and the various lumps that are the rhombencephalon span a pretty good distance. Right in the middle of that is the otic vesicle. It should stick out as a circle. When you get to the actual spinal cord, they give it another name, the myelencephalon. I think they're making this up that you can actually see the forelimb buds on this one, but look for them on your samples. And I will actively tell you not to learn this. So gill slits here is completely improper usage of this, and it gets a lot of evolutionary biologists in trouble when they're arguing with creationists. There are no gill slits on anything but things that have gills. What this is referring to is the pharyngeal arches, which is a defining characteristic of the chordates. We are chordates. They go all the way back to Amphioxus, which is the very first thing to have a head. If you think of all animals, we probably think of the cute furry things. They all have heads. But remember, the definition of an animal is much broader. The chordates are the things that have heads. And the major thing that they invented was something called the neural crest. That is an outcropping from the top of the embryo, the dorsal part, here in the rhombencephalon. And what that does is form all the skull structures, the jaw, as well as adding some of the valves to the heart. It's a very diverse tissue, very radical. But in everything that has ever, after the innovation of this, it forms these arches that creep down and essentially form the face. Okay? And so you'll observe these in embryology, no matter what the species is, so long as it's chordate. And they're called the pharyngeal slits. They are miscalled the gill slits. Human babies do not have gill slits. Human babies have pharyngeal arches or pharyngeal slits. That is how we know a lot of the developmental mechanisms went the same, and there's large discussions about that in terms of evolutionary biology. I encourage you to look at it, but get the terminology right. There's a gross picture. Hopefully you don't see that. Hopefully don't think too much about that.